Today we're going to do the pre-lab for PCR and then the experiment, but this segment is the pre-lab. So hopefully you read your student protocol for the pre-lab and you read the recommended pages in the online manual for PCR. Don't forget you also have PCR in your textbook, and I would highly recommend you reread that. PCR is a pretty complicated experiment, and I really want you guys to understand it. Both PCR and ELISA are used nowadays for testing for infectious diseases. So these topics are particularly relevant in today's world. So PCR, if you hear that they are doing DNA testing for an infectious disease, they're doing PCR. They're taking a patient sample, using PCR to amplify specific regions. I'll slow down and tell you this story in a minute. And then they might be sequencing the PCR results, the reaction result. They might be running it on a gel as we're doing for this experiment. But both PCR and ELISA are new technologies for testing for infectious diseases. Okay, we've set up a story for this experiment to make it so much more relevant for you, and I want to review that with you. So in the scenario we've created, the simulation if you will, there has been an outbreak in a hospital of a particularly nasty strain of E. coli. Remember, everyday E. coli, normal flora found in the colon. We've got a nasty strain of it here. Okay, this nasty strain has been named Bad 1. There's been multiple patients with this infection. Healthcare workers have been using personal protective equipment to isolate themselves and try to slow down the transmission of this nasty strain of E. coli. But this experiment is about trying to determine the source of it. To determine the source, any of the hospital workers that have had contact with the affected patients are being tested. So we are going to be testing simulated DNA from fecal samples. Isn't that nice? From the hospital workers. You're not going to be dealing with any simulated fecal samples, just simulated DNA. We are going to use the DNA to test, we're going to use PCR to test those DNA samples for DNA sequences specific to E. coli bad 1. So we can identify the source and shut down this outbreak. You would be working in groups for this, 3 to 4 students, each setting a couple PCR reactions. But now I'm going to go to the board and introduce you to PCR. I like to talk about PCR as kind of a magic wand. Because, and over here we've got, in case I forget to tell you, PCR stands for Polymerase Chain Reaction. You definitely want to know that for the practical. Now why do I talk about it as a magic wand? Say we've got a tube. I'm going to draw a giant size tube. We've got DNA in it. Say this was from cheek cells. Swab inside a cheek, do a DNA prep, which really isn't too complicated and can be done within an hour. And you end up with a sample of DNA from a patient. In that sample, you're going to have lots of human DNA isolated from the patient's cells. You are going to also have normal flora DNA. Remember? Normal microbiota living in and on us. That's part of us. So you're going to expect human DNA from the cheek cells. You're going to expect normal flora DNA because you've got normal flora in your mouth, which we will look at more in exercise 49. What we're testing for is, do we have DNA from a specific infection? infectious microbe. Okay? So we've got a gamesh of DNA in here. It might not have any of the specific DNA we're testing for, but it might have some. And relatively speaking, it could be a few dots in this gamesh of DNA. So we want to test for the presence of it. How are we going to be able to get in there and do that? That's where PCR is like magic. So PCR was invented in 1983 by Kary Mullis. He did get a Nobel Prize. A key part of the PCR reaction is an enzyme isolated from a microbe that lives in hot springs in Yellowstone National Park. So the discovery of this Taq polymerase is key. It's a DNA polymerase, but a special one. A DNA polymerase that is heat stable. Now, Kary Mullis figured out how to turn this into a PCR reaction, which today is a workhorse in DNA labs. You walk into a DNA lab, they're going to have PCR machines. They're going to have big ones. It's common, it's standard. All right. So if we start with a patient sample, now I talked about cheek cells because it's a little less disgusting, but in our case, in our simulated experiment, fecal samples. You can take a little bit of a fecal sample, do a DNA prep on it, and isolate DNA. So we need a patient DNA sample. Right? So those are provided for us in this kit. Now, what do you need to do a PCR reaction? You need the DNA sample. You need the Taq polymerase enzyme. You need, what are we going to be doing? I haven't talked about that. We're going to be copying a DNA region, very specific to a particular microbe. We're going to be copying DNA, that means we're going to be synthesizing DNA. This is our DNA polymerase. What do we need? What are the building blocks of DNA? We've got to have nucleotides. I might call them DNA nucleotides. In the online protocol, they call them deoxynucleotides. Okay, but we can think of them just as nucleotides. What else do we need? We need buffers, cofactors, anything that is going to create the optimal conditions for the enzyme to work. All right, I've got Dr. Cheney helping me video. I'm going to check in with her. Am I forgetting any ingredients?
Primers.
Oh, key one. I'm going to put them in red. I knew I was forgetting something. Primers. Okay. I am going to do a little drawing on the board here. We are zooming in. I'm going to draw it in black because it shows up better. So this sideways ladder is representing a DNA molecule. And what it's representing is DNA specific for this E. coli bad 1 strain. So here's the research that has to happen before a DNA test. We've got to find DNA that's specific to that strain of that microbe, and we've got to get primers. So the primers are for a specific, unique region of the nasty microbe. or we could say the microbe that's being tested for. Okay? Now, through the magic of PCR, if the specific region of DNA, which is only found in this nasty strain of a microbe, if that is found and amplified in the PCR reaction, then you know that microbe's DNA is in the DNA sample. So that's a positive test for that patient. Okay? Now, to synthesize DNA, we got to open up the strands. All right, this is unit 3 in microbe. I just want to separate these strands out, and we're going to do this. This does occur in a PCR reaction. So we've got a double-stranded DNA molecule. DNA likes to be double-stranded. If you heat it up, the hydrogen bonds between the nucleotides will break open, and you'll get single strands of DNA. At that point, our primers, what the heck are primers? They're short little pieces of single-stranded DNA, right? This is a really short region that I'm doing this for. It'll get us started. And then we'll change to a bigger picture. The primers, short single-stranded pieces of DNA, they mark the region that's going to be copied, amplified by the DNA polymerase in the reaction. Okay? Now, we need a bigger picture. So I want to have a couple much longer. So if I was to put all the nucleotides in, it would take a long time, right? Even longer than that. So the DNA sample, also called template DNA, is that big gamesh of DNA, but we're testing for a very specific region that is specific to the microbe we're testing for. So let's put it in. That's good enough. So the magic is, if this tiny little region, there's just a tiny little bit of it in the DNA sample, if it's present, we can use DNA to find it, okay? So, in a DNA, in a PCR reaction, rather, where do I want to put this? Let's go over on this side. We've got our ingredients, now we'll have our steps. There's 3 basic steps to a PCR reaction. The first is referred to as denaturation. Denaturation. 94 degrees Celsius, that's almost boiling. So everything in the PCR reaction tube gets heated up to 94 degrees Celsius. What happens there? We melt the DNA. It's going to go from being double-stranded to being single-stranded. All right, just single-stranded DNA now with our other ingredients. So in step 2, also known as annealing, the temperature is going to cool down, 50s or 60s Celsius, which is still pretty hot for us humans, but in this picture it's cool. What's going on is that the primers are going to stick. So that's what picture I have up here. The primers are going to stick. It's very specific complementary base pairing, only if they bump into DNA from that E. coli bad one. Okay? If there's nothing complementary, they're not going to find anything to stick to. But if there is, they're going to stick And where they stick, this designates the region that's going to be amplified. So let me write that down. Primers designate, I've said delineate, I've said mark as a simpler word, the region of DNA that's going to be amplified in the PCR reaction. Okay? That's the magic. The PCR reaction can specifically amplify a small region of DNA in this DNA sample, which has all kinds of DNA and multiple copies of it. So that's why I call it magic. You can make primers for a specific region, then you can use PCR. All right? All right. So the primers have bound. Now we'll go on to step 3. Step 3, I'm going to call synthesis. There's another word, Dr. Cheney.
Extension.
Extension. Let's get extension up here too. What's being synthesized? What's being synthesized is DNA, right? What's the enzyme we've got in our tube? All this stuff is in our tube. DNA polymerase. So step 3 is synthesis. The primers have bound. Now we go up to 72 degrees Celsius. These are pretty standard. Annealing temp varies. These are pretty standard. 72 degrees Celsius is the optimum temperature for the Taq polymerase enzyme. Now, I'm going to bring our Taq polymerase enzyme in here as a blue blob. DNA polymerases need a short double-stranded region to start with. You may or may not remember that from lecture, but they do. So that's why we needed the primers. We needed this short double-stranded region. In my mind, it's kind of like a zipper. You've got to get that initial part hooked up and lined up correctly, then you can zip it. So we need this, then the enzyme can come in. I'm just drawing it as a blob. Taq polymerase. And what is it going to do? It's going to synthesize the other strand. Going to have another blob here. Same thing. It's going to synthesize the other strand. I'm not being perfect about matching up all my little nucleotide rungs. So the Taq polymerase has done its thing both ways. You might remember DNA polymerases only work in one direction, but so it's different directions on different strands, but not a major point today. One cycle. We've denatured, we've cooled down to let the primer stick, then we've done the 72 degrees. We've gone from one piece of DNA, double-stranded DNA, to 2. So that's important. One cycle, one cycle of PCR equals doubling the amount of DNA. So, these 3 steps are done over and over and over again. Right?
You spelled cycle wrong.
I spelled cycle wrong. Oh my goodness, let me fix that. That could be very distracting. Okay. Chain reaction, we're doing this over and over and over again. So we're using a polymerase. We're doing the cycle of temperature changes. Each cycle, we double the DNA. But is it any DNA? No. It's the specific region that we're testing for. What delineates or specifies that specific region? The primers. Short single-stranded pieces of DNA. So where they stick, that's where the Taq polymerase can come in, synthesize the other strand, and we went from 1 copy to 2. Now, if you want to see a lovely picture of that, look at the online protocol or look at your textbook. I am not going to draw one for you on the board because that would be crazy. But if you start with 1 piece at the top, 2 pieces, you keep doubling, 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 you get a crazy amount of this little region of DNA. So that is the plan. So this section is going to take you through setting up the PCR experiment. The tools we're going to be using micropipettes, and they are very precise. Not like the pipette you were using for that plate count experiment. So these come in a set of 4. The ranges, I'll turn it this way. How are we doing? 0.5 to 10 microliters. A microliter, there's 1,000 of them in a mil. 20 to 200 microliters. 5 to 50 microliters. Dang, they're not in the right order, but we'll fix it. It's all good. 0.5 to 10, 5 to 50, 20 to 200, and 100 to 1,000. So this is the range you should use. Don't try to use them for anything outside of that range. Number 1. Number 2, use the smallest range possible to be the most accurate. So if you're pipetting anything 10 or below, you want to use the gray top. It'll be the most precise. You could do 6, 7, 8, 9, 10 on here, but don't do the orange top if you can do the gray top. For today's lab, we're just going to be using the gray and the orange, okay? If you were here practicing with them, you'd also be using the 20 to 200, but we're not. So that's a set of them. The range is here. Now, turn it to the side, the number, the exact microliters. You turn the colored part at the top. I'm going to put this to 20 because I think that's one of the volumes we're going to be using. So when you're ready to use it, you set the volume, okay? And we'd be walking through this together anyway. So I'd tell you if you're using the gray or the orange. Set the volume. Now, how do they actually work? We have disposable sterile tips. So you put a disposable sterile tip on it, and then hold like so. Most people, the thumb's the strongest. This is the motion for sucking up the liquid and for spitting the liquid out. So similar to those pipettes we use for the plate count, push this down, stick the tip in the liquid, release, it sucks up the liquid. Now move your tip to where you want it to go, push all the way down to spit the liquid back out. Trickiest part is when you're going down, getting ready to suck up the correct volume, you go down to where the first resistance is. Okay, that's the hard part. So down to where you first feel that resistance, hold it there. But when you're spitting out, you go all the way down. Okay, so these are micropipettes, sterile disposable tips. We're going to have a trash beaker here. So things are going to go in the trash beaker. And now that I've walked you through the pipettes, Dr. Cheney and I are going to set up PCR reactions, and you get to watch since you don't get to do them yourself. So we're going to be looking at the student protocol, which hopefully you printed and read already. So we've got, these are called Eppendorf tubes, these little tubes, Eppendorf racks. These you can actually get on Amazon. And let me show you the equipment we've got out. So what do we need? We have an ice bowl because some of these reagents should be on ice while they're being used. We have a microcentrifuge. We can put in 6 of the little tubes and spin them to get all the liquid down to the bottom. We've got a beaker for trash. We've got pipettes, we've got tips, and then we've got our tubes in here. We've got tubes with water, tubes with primer, tubes with our samples we're testing, and tubes with positive and negative controls. These little tiny tubes are what we're actually going to be setting our PCR reactions in. Okay, each group, if you were here, would have half of this rack and be setting 2 PCR reactions. Okay, and Dr. Cheney and I are going to set up 4 sets of reactions for the video. So, I think we're ready to start. Are you ready? Okay. Oh, we have to label first. Tiny PCR tube with the white ball in it. The white ball has the polymerase and other good stuff for this chemical reaction. So with the white ball in it, it's number 1. We're also going to do a group symbol. I think I'm going to do a circle. So that's number 1. Put the group symbol on both of these. The second tube is 2. So I'm putting a 2 and a circle. Right. For the small volumes, we can actually just lay them on their side. The liquid will stay in them. Okay, we're on to step number 3. We're going to add 20 microliters of primer mix, which is a yellow liquid. For 20, I'm going to go with the orange top pipette. It's set at 20. What do you know? Tip is firmly on. Oh, you know what? I got to do both of these. So what symbol did you use?
I used a star and just a dash.
Star and a dash. So I'm going to use a triangle. I forgot I had to set both. Oh dear, look at that. Little stinkers. The static electricity, the gloves. Number 1, number 2 can cause that to happen. Okay, now I'm actually ready for step 3. Into tube 1. Tubes 1s have the little balls of dehydrated enzyme and other good stuff. 20 microliters of primer mix. So we open the little tubes. You might be able to see just that little bit of water in there. I am going to depress to the first resistance, stick the tip in the water, release, and it sucks up the wire. I'm now done with this tube. The tube goes in the trash, the water goes into tube number 1. And that white ball is magically going to disappear because it's dissolving in the water.
You ran water first?
Oh, shoot. Oh, yeah, we're all good. It's supposed to be
I just wanted
primer
to make
mix.
sure I wasn't doing something.
Oh, you're doing fine.
All right.
Primer, yellow. We'll do that. Okay. Primer to my second number 1 tube. So the primer was 20, water was 15. Oh, I don't want to close it. There's 2 ones. So now I'm doing 15. I'm going to use the same orange top. Dial it to 15. Now the water tube. So the white ball has water and primer. So now, step 4, make sure the lids are tightly closed. And flick to mix. This is what it looks like. I'm going to make sure that white ball is totally dissolved and everything is mixed well. I'm going to do that another time. Flicking tube. We'll spin all 4 of our tubes together. Literally 2 seconds does the trick. This is to make sure all the liquid's at the bottom of the tube. Now, we're going to pop them on ice. Okay. Alrighty. And proceed to step 5. Now we are removing 19 microliters from tube 1 into tube 2. I gotta pay attention. Let's see, I'm over here. Triangle. 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 For this one. We covered that steel too. Okay. Now I'm on my circle. 19 microliters. Try to stick them in pairs in there, but one just slipped onto the bottom. I'll be sorry. Okay. Next, we're adding samples to the tubes. Okay, so I'm going to take 2 at a time out. So I'm going to have, make a little note here, circle, triangle. I'm going to record in my notes which is which, because 2 tubes are going to get patient samples. The patient samples are letters A, B, C, and D. Dr. Cheney has A and B. I've got C and D. The other 2 tubes are going to get controls. I have a positive and a negative, a P and an N, and so does Dr. Cheney. So if I pull out, these are my triangles. Oh, the other thing is don't put the tiny little tubes in the rack. They're hard to get out. Maybe harder for some people than others. So triangles are on this side. Tube 1 will be the sample. So tube 1 is an A, B, C, or D. I'm going to give it the D. And how much? 2.5 microliters. We've got to go down to the gray top, down to 2.5. So, triangle, I'm doing D. If you could see this tiny little volume. That's all it is in the pipette tip. D. And that goes in tube number 1. It is very important to keep track of what's going where, so that when you run your samples on a gel, you know what the heck is in each sample on the gel. So tube number 2 for this one is going to be a P, positive control. Tube 2. Now, more recently, they added color to some of these solutions. So that if you got all your ingredients, you should come up with orange, because we had a yellow solution and a red solution. There's 2. Now I got my circles, 1 and 2. So 1, C, so this is the person sample. Goes in 1. Now the control, this time the N, is going into 2. Page, flip and spin, I do believe. Your protocol says it should be green. They changed colors on us. Now it should be orange. Okay. So Dr. Cheney spun hers down. I'm going to spin mine down. Then we're going to walk over and put them in the PCR machine. Some paper and a pen. So you can write down what order we put them in the machine, because we've labeled them.
I'm sorry.
We've labeled them, but
And negative, and then the star is being positive. Yes.
whoa, okay. Take our 4 tubes over. So this is the thermocycler. I'm gonna put the little tubes in there. But I'm gonna make a plan here. Circle 1, circle 2, triangle 1, triangle 2, and put them in that order. Circle 1, circle 2, triangle 1, triangle 2. And then Dr. Cheney's going to do a similar thing.
So it's star 2. Okay.
Alrighty. So they're in place. This metal part is the temp block that's going to be changing temperatures for the PCR reaction. So in a different segment, I'm going to talk about PCR and everything that's going on. But this is the temp block that will be changing temperatures, because we're going to polymerase chain reaction. PCR is a cycle, and it's a cycle of temperatures. So this machine is going to do that cycling for us. And it's pretty nifty. Program's already in there. Looks like we got a preview. And I think we just want to press run. Our volume. Good. And now you get to see in a picture form. There's going to be an initial longer denaturing step at 94 degrees. That's just at the beginning. Steps 2, 3, and 4 are the steps that are going to be repeated over and over and over again. So denature. Cool down to 45 degrees. That's annealing. That's when the primers are going to stick to the single-stranded DNA template. And then up to 72 degrees, that is the optimal temperature for the Taq polymerase enzyme. And then over again. So when you get up to around 30, 35 cycles, your product plateaus. So typically you do between 30 to 35 cycles. In this one, we're doing 30 cycles. At the end, there's a long extension step, 5 minutes. And then the machine's going to go down to 12 degrees and hold it there until someone comes in tomorrow and gets the tubes out. PCR reaction, typical time, 2 to 3 hours. The time remaining is showing right here. So this reaction has got less than 2 hours to go. Okay? That's it for today.